Hello, science enthusiasts, and welcome back to the channel. Today, we're embarking on a journey back in time to the dawn of a new millennium to witness one of the most ambitious and monumental quests in the history of science. Forget climbing Everest or landing on the moon. This was a voyage inward, an exploration of the most intimate and complex landscape imaginable, the human genome. We're talking about the Human Genome Project, the audacious international effort to read, letter by letter, the entire genetic instruction book for a human being. It was a project of staggering scale, promising to unlock the deepest secrets of life, disease, and what it truly means to be human. So buckle up, because this is the story of how we decoded the blueprint of humanity itself. To truly grasp the magnitude of the Human Genome Project, or HGP, you have to understand what it was up against. Imagine a book, or rather an entire library, written in a language with only 4 letters, A, T, C, and G. These are the nucleotide bases, adenine, thymine, cytosine, and guanine. This library contains the complete instructions for building and operating a human being. Now imagine this library contains not a few thousand or even a million, but 3 billion of these letters. That's the human genome. 3 billion base pairs, coiled tightly into the 23 pairs of chromosomes found in nearly every one of our cells. Before the HGP, we could only read tiny fragments of this book, like finding a single, isolated word in a vast, unindexed library. The goal of the project, launched in 1990, was to read the entire thing from cover to cover and create the first ever reference map of our genetic code. This wasn't just a scientific curiosity, it was a mission driven by the promise of revolutionizing medicine. Scientists knew that hidden within these sequences were the genetic culprits behind diseases like cancer, cystic fibrosis, and Huntington's disease. By mapping the genome, they hoped to find these faulty genes, understand how they worked, and develop new ways to diagnose, treat, and even prevent them. The project was an unprecedented international collaboration involving scientists from the United States, the United Kingdom, France, Germany, Japan, and China. It was a testament to what humanity can achieve when it works together, a truly global effort to understand our shared heritage. But like any great epic, this story has its heroes and a dramatic rivalry that pushed the boundaries of what was thought possible. On one side, you had the publicly funded international consortium, led by the thoughtful and determined Dr. Francis Collins. Their approach was methodical, meticulous, and public. They were mapping the genome piece by piece, chromosome by chromosome, ensuring accuracy and making all their data freely and immediately available to the world. It was science for the public good, a slow and steady march toward the finish line. Then, in 1998, the race was thrown into chaos. A brilliant, controversial, and fiercely competitive scientist named J. Craig Venter entered the ring. Venter, backed by private funding through his company Celera Genomics, announced that he could sequence the entire genome much faster and cheaper. He planned to use a new, more aggressive technique called whole genome shotgun sequencing. To understand the drama, you need to understand the methods. The public project's approach was like meticulously translating a book, one chapter at a time, making sure every sentence was perfect before moving on. They would map large sections first, then break them down to be sequenced. Venter's shotgun sequencing, on the other hand, was like taking 100 copies of the book, shredding them all into tiny overlapping fragments, and then using supercomputers to piece the entire story back together based on where the fragments overlapped. It was a high-risk, high-reward strategy. Many in the public consortium thought it was reckless and would be riddled with errors. Venter bet his reputation and his company on the idea that computational power could solve the puzzle. This ignited a fierce race to the sequence. It was the public, collaborative, open-source approach versus the private, fast-paced, proprietary model. While the media loved the drama, this competition had a profound effect. it accelerated everything. Both teams worked at a feverish pace, generating data faster than ever imagined. The technology improved at an exponential rate, and the cost of sequencing plummeted. The race, while tense, ultimately benefited all of humanity by getting the data out years ahead of schedule. On June 26, 2000, in a joint announcement at the White House, President Bill Clinton, flanked by both Francis Collins and J. Craig Venter, declared the completion of the first rough draft of the human genome. It was a landmark moment, a scientific ceasefire that marked the end of the race and the beginning of a completely new era. The book of life was finally open. But mapping the genome was never the end goal. It was the starting point. The real question was, now that we have the code, what do we do with it? This single question would unleash a revolution in medicine and simultaneously open a Pandora's box of ethical dilemmas that we are still grappling with today.
The completion of the Human Genome Project wasn't an endpoint. It was the firing of a starting pistol for a new kind of biological and medical research. With the 3 billion-letter reference sequence in hand, scientists now had the ultimate roadmap to explore human health and disease. The impact was, and continues to be, utterly transformative, ushering in an era we now call personalized medicine. Before the HGP, medicine was largely a one-size-fits-all affair. A diagnosis would lead to a standard treatment, and doctors would hope it worked. But we all know people who react differently to the same medication. The HGP gave us the why. It opened the door to pharmacogenomics, the study of how your specific genes affect your response to drugs. Today, doctors can test for specific genetic variations to predict whether a patient will have a bad reaction to a certain anesthetic, or to determine the exact optimal dose of a blood thinner like warfarin. For cancer treatment, this has been a game changer. Instead of just carpet bombing the body with chemotherapy, doctors can now sequence the tumor's genome, identify the specific mutations driving its growth, and select targeted therapies, drugs designed to attack only the cancer cells with that specific vulnerability. This is precision medicine, and it's a direct legacy of the HGP. Beyond treatment, the project revolutionized diagnostics. For the first time, we could definitively link specific diseases to specific genes. Take the BRCA1 and BRCA2 genes. Mutations in these genes dramatically increase the risk of developing breast and ovarian cancer. Thanks to the HGP, we can now test for these mutations, allowing individuals with a family history of cancer to understand their risk and make informed decisions about preventative measures, like increased screening or prophylactic surgery. This power extends to countless other conditions, from single-gene disorders like cystic fibrosis and Huntington's disease, which can now be identified with near-perfect accuracy, to complex conditions like heart disease and diabetes, where we can identify dozens of genetic factors that contribute to an individual's overall risk. We've moved from treating diseases after they appear to predicting and potentially preventing them before they even start. This knowledge is power, the power to take control of one's own health destiny. But this newfound power came with an immense and profound responsibility. Perhaps the most forward-thinking aspect of the Human Genome Project was that its leaders anticipated this. From the very beginning, they dedicated a significant portion of their budget, around 3 to 5%, to an unprecedented program called ELSI, which stands for Ethical, Legal, and Social Implications. They knew that reading the human genome would raise some of the most challenging questions our species has ever faced, and they wanted to start the conversation immediately. The first and most obvious concern is genetic privacy. Your genome is the most intimate, personal information you possess. It contains clues not only about your current health, but also your future risks for developing certain conditions. So who should have access to it? Your doctor? Okay. Your health insurance company? What if they use it to raise your rates or deny you coverage based on a predisposition to a disease you may never get? What about your employer? Could you be passed over for a promotion because a genetic test suggests you might be at higher risk for a neurological condition later in life? This fear of genetic discrimination was so real that it led to the passage of laws like the Genetic Information Non-Discrimination Act, GINA, in the United States, which makes it illegal for health insurers and employers to make decisions based on genetic information. But as data becomes more widespread, the questions of data security, consent, and ownership are more pressing than ever. And the ethical questions only get deeper. The HGP gave us the map. Newer technologies, most famously CRISPR-Cas9, have now given us the tools to edit that map. This opens up incredible therapeutic possibilities, the potential to correct the genetic mutation that causes sickle cell anemia or Huntington's disease at its source. This is gene therapy, and it's a dream come true. But it also opens a path to gene enhancement. If we can fix a bad gene, what stops us from improving a normal one? What's the line between preventing disease and enhancing intelligence, athleticism, or appearance? This leads us down the path to designer babies and raises terrifying questions about creating new social divides, a genetic upper and lower class. Where do we as a society draw the line? Ultimately, the Human Genome Project forced us to confront a fundamental philosophical question. What does it mean to be human? Are we simply the sum of our A's, T's, C's, and G's? The project's results actually provided a comforting answer. No. We learned that the genome is not a simple deterministic blueprint. It's an incredibly complex, dynamic script that interacts constantly with our environment, our choices, and with chance itself. It revealed that genetically, all humans are 99.9% identical, a powerful scientific testament to our shared humanity. The Human Genome Project didn't just give us a map of our DNA, it gave us a mirror, forcing us to look at our science, our medicine, our society, and ourselves in a new and much more complex light. The story of the Human Genome Project is the story of a monumental scientific achievement that continues to unfold every single day in labs and clinics around the world. It was a quest that gave us the tools to fight disease and understand life in ways our ancestors could only dream of. But it also handed us a profound ethical torch, one that we must carry forward with wisdom, caution, and a deep sense of our shared responsibility. The code has been read, but the story of what we do with it is still being written by all of us. Thank you so much for joining us on this incredible journey today. What do you think is the most significant impact of the Human Genome Project? Let us know in the comments below. And if you enjoyed this deep dive into the science that shapes our world, please don't forget to hit that like button, share this video with a friend, and subscribe to the channel for more content like this. We'll see you in the next one.